Hello, everyone, and welcome to today's webinar, Novel Therapeutic Approach to Lethal SARS-CoV-2 Infection in Aged Marine Models. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotech. To learn more, visit them at miltenybiotech.com. Now, we encourage you to participate today by submitting any questions you may have during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd now like to welcome our speaker, Dr. Jian Zheng, Assistant Professor in the Department of Microbiology and Immunology at the Center for Predictive Medicine at University of Louisville School of Medicine. Dr. Zheng, welcome. You may now begin your presentation, sir.
Thank you, Susie. Thank you for your nice introduction. And it's my pleasure to get an opportunity to introduce our work about the SARS-CoV-2 infection, especially in the mouse models. So let's just start. So most of you may have never heard about coronavirus before this pandemic, but actually the infection of coronavirus in human population can be traced to 1,000 years ago. And around 100 years ago, another coronavirus variant called OC43, which belongs to the same class, just like the SARS-CoV, MERS-CoV, and SARS-CoV-2, was found in the human population, which caused some flu-like symptoms, but it's not severe. More recently, another coronavirus variant called SARS-CoV was reported in the Guangdong province in China in November 2002. SARS-CoV had spread globally by July 2003, and this coronavirus caused around 10% mortality. So it can cause very severe diseases, but the transmission of this virus is kind of limited, so it disappeared after that, and no one knows why it just disappeared like nothing happened. Around 10 years later, another coronavirus called MERS-CoV was isolated from the spreading of a 60-year-old Saudi Arabian, and this coronavirus was still known as the most lethal coronavirus in the world because it can cause 30% mortality in the hospitalized patients. And then we face the most severe global pandemic caused by the SARS-CoV-2. Just like MERS and the SARS-CoV, it's still a single-strand RNA virus. And till today, this pandemic has not disappeared. And we don't know when it will end, but what we know is that it has caused more than 6 million deaths in the whole world. And with the more and more variants occurring from this original type of the virus, it looks like the vaccine or the previous infection cannot stop the spread of the new variants completely. And we hope it will end as soon as possible. Although the SARS-CoV, MERS-CoV, and SARS-CoV-2, all of them were regarded as transmitted from the bats or from some other animals by the natural reservoir to the human population. But these three different viruses can enter the human cells via the different receptors, such as the human DPP4 and the human ACE2. The entry of this virus into the human bodies can cause some mild to severe symptoms of the patient. And another common characteristic of these three different coronavirus infections is that in the older population, it can cause severe diseases. So, to find out whether there are some aging-related factors contributing to the age-related diseases severity caused by the coronavirus, our lab just used an animal model to try to find out the underlying mechanisms. The first thing we did was to compare the young and middle-aged mice, their responsiveness to the SARS-CoV infection. Because when we started this project, we still have no SARS-CoV-2. It's like almost 20 years ago. So what we did is that we infected young and middle-aged mice with the SARS-CoV, and we found that just like human, the middle-aged mice shows very sensitive to the SARS-CoV infection. And you can see in this figure, the middle-aged mice, they all died within 12 days post the infection, while the young mice, just none of them was dead. So we want to know what is the difference between the young and the middle-aged mice. So we sacrificed some young and middle-aged mice and then isolate their lung to run the RNA microarray. And we found that compared to the young mice, the expression level of PLA2G2D in the middle-aged mice was increased significantly. And the PGD2, which is the downstream product caused by the PLA2G2D, was also increased in the middle-aged mice. And this difference will be even increased after the infection of the SARS-CoV. So it looks like the PLA2G2D and PGD2 contribute to the age-related diseases severity in SARS-CoV infection. Now, you may ask, what is the PLA2G2D and PGD2? So before we just continue our story, let me just give a brief introduction about this signaling pathway. PLA2G2D is belongs to a phospholipids superfamily, and this superfamily are composed of the multiple enzymes. These enzymes can hydrolyze the phospholipids into the long-chain fatty acid, and this long-chain fatty acid could be further catalyzed or oxidized by the different enzymes to generate the multiple different lipid products, and including the PGD2, prostaglandin D2, PGE2, prostaglandin E2, and a lot of other molecules. In our lab, we focus on the PGD2. PGD2, prostaglandin D2, could bind to the two different receptors in different human cells and also mouse cells. It can bind to two major receptors, DP1 and DP2. The DP2 was mainly expressed in the T cells and was regarded to induce the Th2-related inflammation such as the asthma and the allergic reactions. And DP1 mainly expressed in the myeloid cells. The binding of PGD2 and DP1 can induce the increased production of CAMP, which will lead to the inhibition of the inflammation and further to decrease the secretion of the pro-inflammatory cytokine like the IL-1 beta. And we all know that IL-1 beta has been regarded as a major mediator contribute to the SARS-CoV and SARS-CoV-2 related diseases. So, since the expression level of PLA2G2D and PGD2 increased with the age, and the SARS-CoV also is age-related diseases, so we think that maybe this signaling pathway was involved in the SARS-CoV infection-related pathogenesis. To clarify their roles, we need some model, and we are lucky to find some animal model. Firstly, we got a PLA2G2D knockout mice model from our Japanese collaborators. And when we challenge this PLA2G2D knockout mice and compare them with the wild-type mice, we can see in the left panel. The wild-type mice, which also marked as PLA2G2D double-positive mice, they were still susceptible to SARS-CoV infection. But if this molecule was knocked out in the mice, they were resistant to the lethal challenge of SARS-CoV. Consistent with that, the virus clearance was increased in the PLA2G2D knockout mice. So PLA2G2D's role was confirmed. And our next question is that whether DP1 knockout can also show the similar phenomenon. So we got another knockout mice, DP1 knockout mice from another Japanese collaborator. And here in the left panel, you may see that, okay, it looks like not much difference in the weight change between the wild type and the DP1 knockout mice. But you can see in the right panel, the DP1 knockout mice, they provide a resistance to the lethal challenge of SARS CoV compared to the wild type mice. So, taken together, these results suggest that PLA2G2D and DP1 signaling may contribute to the acute SARS CoV infection. Now let's go back to the pandemic, to the COVID-19. As we mentioned previously, like MERS-CoV and SARS CoV, COVID-19 is also a disease of the elderly. Based on the clinical data, we found that if the people was older than the 65 years, the rate of hospitalization and the rate of death will increase significantly. So we want to know that whether the PLA2G2D and PGD2 also involved in the human SARS-CoV-2 infection. But before that, we need some animal model. As you may know, the normal mice were resistant to the SARS-CoV-2 because these mice was lack of the receptor to the SARS-CoV-2. In the human, the SARS-CoV-2 need to enter the cells via a receptor called human ACE2. But mice lack of this receptor. So the first thing we need to do is to create some mice model can be infected by the SARS-CoV-2. The first model created by our lab is the 85 human ACE2 model. And this model can be established in any normal mice because it will just transgenic the human ACE2 into the mice. The benefits of this model is that mice are sensitized for the SARS-CoV-2 infection by transduction. It's a very simple manipulation. And these strains can be directly assessed without additional breeding. These mice are also useful for determining the host factors necessary for optimal virus clearance and for the assessing efficacy of vaccines and therapies such as the convalescent plasma. However, this model has a very major shortcoming. This shortcoming is that this mice model can only develop very mild diseases after challenged by the SARS-CoV-2. So it cannot mimic the severe case in clinic. To establish some models for the severe case, our lab introduced the second model. It's called K18 model. And in this model, the mice will be express human ACE2 after the promoter called K18. As you can see in the right panel, these mice can develop mild to severe diseases based on the different dose of the virus inoculated. So it can mimic the mild to very severe diseases, and some mice can be killed by the virus challenge. However, this model also has a major shortcomings because if you want to investigate the role of specific molecule like the PLA2G2D and PGD2, you will need to cross-mating the K18 mice with the knockout mice, and it's very time-consuming and effort-consuming and money-consuming. Since the pandemic spread so fast, we need some more quick method. So that's the reason we try to establish the third model in our lab, which was called mouse-adapted virus model. And we know that we already have found a lot of different variants of the SARS-CoV-2 apart from the original strain. So in these different strains, the virus founded in the South Africa showed a single mutation called N501Y in the spike protein. This single mutation can increase binding affinity between the virus spike protein and the mouse H2. So it can help the virus enter mouse cells. But this entry of the virus can still not cause the significant diseases in the mice. So our solution is to use the reverse genetics to introduce this single mutation into the original SARS-CoV-2 virus and then passage this virus in the Barbacin mice from P0 to 30 passages. Briefly, the method is very simple. We just use the mutated virus to infect the mice, and then after 2 to 3 days, we will sacrifice the mice and harvest the lung to make the homogenate and use this homogenate to inoculate the next generation of the mice. So after 30 passages in the mice, this virus became very virulent. And more interestingly, this virus, in this virus, we used the sequencing to identify the mutation in this virus, and we found that just like in human, this passage can favor the more mutation development in this virus, and all this mutation can be found in the human SARS-CoV-2 variants, which means that actually there's not too many difference between the human and the mouse virus passage. So after getting this virulent virus, here you can see that this virus can kill both male or female Barbacin mice, and no matter they are young or old. But this virus can only kill the older B6 mice, but not young B6 mice. And in addition to their susceptibility to the virus challenge, we also found that the infection of these mice can induce both the antibody, virus-specific antibody and the virus-specific T-cell responses. And here we use the peptide pool provided by Miltenyi Biotech. And we found that after challenge with the spike protein peptide, M protein peptide, and membrane protein peptide, we can find that CD8 T cells isolated from these mice can secrete high expression of interferon gamma. And this taken together, all of this data suggests that this model can mimic what happened in the human patient. Since we found that the mouse-adapted virus can kill the old mice but not young mice, then we got a perfect model for the age-related diseases, which can be also found in the human patient. So with this model, we are again use the PLA2G2D knockout mice and the DP1 knockout mice to test the role of this signaling pathway in the SARS-CoV-2 related diseases. And here you can see that the knockout or deficiency of the PLA2G2D or DP1 can offer the complete or incomplete protection on the mice infected by the SARS-CoV-2. But of course, these mice are 7 to 8 month old mice. And for the young mice, 8 to 10 weeks old B6 mice, they were resistant to the SARS-CoV-2 virus. So here, this data suggests that the PLA2G2D and DP1 signaling pathway could be a potential therapy targets for the human SARS-CoV-2 infection, especially in the elderly. Consistent with their benefits, the deficiency of the PLA2G2D or DP1 will also decrease the viral load in the infected lungs and ameliorated the pathogenesis in the lung of the mice infected with SARS-CoV-2. Now, after confirming the benefits of the PLA2G2D and DP1 knockout in this mice model. The next step is to find out whether we can get some compounds or some drugs to targeting at least signaling pathway to treat the SARS-CoV-2 infections. Unfortunately, we cannot find the optimal candidate for targeting PLA2G2D, so we have only focused on the DP1 targeting therapy. But actually, it's not a totally bad thing. It's actually some kind of the good things because it can narrow the efficacy of these drugs, and it will be more specifically because we know that at the downstream of PLA2G2D, not just the PGD2 can be generated. So by working with a company called BioAge, we got a compound called Asapiprant, which is a DP1 antagonist. And the benefits of this drug is that it has been passed the phase 1 clinical trial because it was developed several years ago to treat some autoimmune diseases, including the arthritis. So it's safe, it's ready to use. So here we just got a compound from the company, and then we designed the experiment. We will infect mice with SARS-CoV-2 or SARS-CoV, and then start the treatment at the day 2 or day 4 post-infection. The reason we choose a later treatment is to try to mimic what we found in the clinic, because generally the patient infected by the SARS-CoV or SARS-CoV-2 will underline some like the latency period before the onset of the symptoms, and it doesn't make sense to treat the patient or treat the mice before the onset of any symptoms. So we give the treatment, and the outcome is really exciting. It's even beyond our expectation. In this figure, you can see that almost all mice treated with the Asapiprant were protected from the lethal challenge of SARS-CoV-2. And the treatment of Asapiprant also decreased the viral load in the lung and improved the pathogenesis in the lung. So again, this DP1 targeting treatment looks like a promising candidate for the future treatment against the SARS-CoV-2. But also, we want to find out how the DP1 antagonist can benefit the SARS-CoV-2 infections. And we try to just go through about the signaling pathway to find the answer. The first thing we did is to try to find another candidate in this signaling pathway to see whether some other molecules also be involved in the benefits mediated by Asapiprant. So here you can see that from PLA2G2D to PGD2, they need to be manipulated by an enzyme family called COX-1 and COX-2, which has been, the COX-1 and COX-2 inhibited has been widely used to treat a lot of diseases like fever, like the inflammation, like pain. And Indomethacin is one of the most representative drugs in this family. So we just used the Indomethacin to treat SARS-CoV-2 infection. And we found that, okay, it looks like it did not work well. It's kind of disappointing because the simplest way did not solve the problem. But it's understandable because as I mentioned previously, at the downstream of the PLA2G2D signaling, there's a lot of different lipid products, and PGD2 is just one of them. And there's another like the PGE2, PGF2 alpha, and a lot of things. And these products, some of them may be pro-inflammatory, some of them may be anti-inflammatory. So if you block the upstream, it might not show anything. And also, since the PLA2G2D and PGD2 expression level will be increased with the aging, so Asapiprant, when we treat the young mice, they did not affect the SARS-CoV infection in these mice. But here I have to mention that since the young mice are kind of resistant to the SARS-CoV-2, so they did not show much severe diseases as the older mice. And here you can see they only showed some like the milder weight change. So here we can only say that Asapiprant does not affect in the young mice in our model, but to the human population, the efficacy needs to be investigated in the future. Now, after confirming their roles, we want to know more about the mechanism. And in our previous studies, we found that the blocking PGD2 function with the small molecular antagonist will enhance respiratory dendritic cells migration, T cell responses, and the survival in the SARS-CoV infections. And we just wonder whether this mechanism also contributes to the Asapiprant-mediated benefits. So here we just stain the respiratory dendritic cells by a non-specific dye called CFSE. And then after infection, we found that the infection will push some of the respiratory dendritic cells to the drain lymph node to induce the T cell mediated immune responses. And with the treatment of Asapiprant, the migration of the dendritic cells from the respiratory system to the drain lymph node will be increased. So it looks like the increased migration of dendritic cells from lung to the drain lymph node is contributing to the Asapiprant-related benefits, but it's just one of them, and we don't know whether some other factors can be involved in this process, and the study is still ongoing. Also, we found that in the blood, the Asapiprant treatment will increase the T cells accumulation, but also decrease the neutrophil accumulation. The similar phenomenon could be also found in the lung. And current clinical data suggests that the neutrophil may play an important role in the pathogenesis induced by the SARS-CoV-2. So maybe this mechanism also play a role in the Asapiprant-mediated benefits. We also try to find out some other serum and the lung cytokine or chemokine expression to see whether they could play a role in the pathogenesis of the SARS-CoV-2. But the information are limited. It's not like a huge. But we also saw that the CRP protein and the IL-6 and CSL1 was decreased with the Asapiprant treatment, which again suggests that the DP1 targeting therapy could ameliorate the inflammation caused by the SARS-CoV-2. And finally, after we confirm the role of this treatment in the SARS-CoV-2, we go back to the SARS-CoV infection model, and we found that yes, this Asapiprant treatment can not only work in the SARS-CoV-2, it can also work in the SARS-CoV. It's not surprising because if you still remember in my first several PowerPoints, you can see that the PLA2G2D and DP1 knockout mice are resistant to the SARS-CoV infection. So in summary, here we just show that to find a solution for the global pandemic, we firstly established some mouse SARS-CoV-2 infection model, and then we found that the knockout of the PLA2G2D and DP1 could completely or partially rescue mice from the lethal challenge by the SARS-CoV or SARS-CoV-2. And then in the next step, we found the DP1 antagonist Asapiprant can be used to protect mice from lethal challenge of this coronavirus infection. The benefits of this drug was mediated by the increasing the lymphocyte and decreasing neutrophil accumulation in the lungs. So far, everything looks perfect, and we found a novel therapy against the coronavirus infection, and that's perfect. But it's not all the story. In another study carried out by our lab, we found that the Ph.D deficiency was not always beneficial. In this model, we used the wild type and the Ph.D knockout mice, and we established a MERS-CoV infection model in these mice. And in this model, we already know that the knockout mice, they can show the resistance to the challenge. Here you can see when we compare the naive HDPP4 mice, we use the HDPP4 knock-in mice because it's a receptor for the MERS-CoV. So without this receptor, mice will be resistant to the MERS-CoV infection. So when we compare the naive HDPP4 mice and the naive HDPP4 PLA2G2D knockout mice, we can see that the PLA2G2D knockout mice, just like in SARS CoV and SARS CoV-2 infection, they were resistant to the MERS-CoV challenge. However, after they were treated with the sublethal dose of the MERS-CoV, which means this challenge will not kill mice, but it can favor the generation of virus-specific immune memory in these mice. And theoretically, this immune memory can protect mice from the secondary challenge of the same virus. And here, if you compare the naive and the immunized HDPP4 mice, you can see that the immunized mice were protected completely from the secondary challenge. However, if they are knockout mice, the immunized and the naive mice did not show much difference when they were challenged with the same virus again. This data suggests that the immune memory they are not good in these knockout mice. So to find out why, we then harvest some samples from the wild type and the knockout mice. And we also use to exclude the sensitivity to the infection. We also use a vaccine to immunize the mice and then challenge them with the live virus. And the result is the same. The knockout mice did not generate the effective immune memory against the second challenge of the MERS-CoV. Not just the intranasal immunization, but also the IP, the intraperitoneal immunization, also could not induce a good memory in these mice, but it is improved in the knockout mice. So again, this deficiency caused by the PLA2G2D knockout in the immune memory was more related with the respiratory tract immune system, which means that there must be something wrong in the airway happened in the knockout mice, which resulted in the less efficient immune memory generation. Since the virus-specific neutralizing antibody is the major weapon used by the host to fight against the secondary challenge of the same virus, our first step is to compare the antibody production in these knockout mice and the wild-type mice. And here you can see that, especially in the middle-aged mice, if they were immunized via the intranasal manner, and you can see that the knockout mice cannot induce efficient amount of the neutralizing antibody. However, in the young mice, there's no significant difference between the knockout mice and the wild-type mice antibody titer. And if the mice were treated with IP immunization, the antibody level is also comparable. So again, since the PLA2G2D and PGD2 expression level was increased with the aging, so all this data suggests that the aging in this knockout mice, there must be something happened in the respiratory immune system to cause the less antibody production. So since the B cells was the major, or you can say almost the only origin of the neutralizing antibody, so we want to know that besides the antibody difference, whether the B cells also show some difference in these different mice, in the knockout between knockout mice and the wild type mice. And here you can see that the total B cells and even memory B cells doesn't show any difference between the wild type and the knockout mice. However, the knockout mice show the less plasma cells, which is the major antibody secreting cells, and also germinal center B cells. These germinal center B cells are mainly a region of the plasma cells in the secondary challenge. So it seems that Ph.D deficiency doesn't affect the whole B cell population. It just affects the antibody producing B cells. So the B cells were impaired, but how about T cells? When we compared the CD4 and CD8 T cells in the knockout mice and the wild type mice, we found that the Ph.D deficiency will increase the production of TNF, alpha, and IL-2, but decrease the secretion of IL-21 in CD4 T cells. And the TNF and IL-2 are mainly known as correlated with the inflammation in the SARS, MERS, and SARS CoV-2 infection. And IL-21, what is the role of these cytokines? Okay, if you are familiar with the immunology, you might realize that the dendritic cells get the antigen and then presented to the CD4 T cells. And there's a specific population of CD4 T cells called the follicular T cells. The major role of these CD4 follicular T cells was to induce the B cells into antibody producing B cells. And one of the major signal transfer from the CD4 follicular T cells to the B cells or to itself themselves is IL-21. So IL-21 is a critical role cytokines for the development of follicular T cells and also antibody producing B cells. The decreased expression of IL-21 by CD4 T cells is correlated with the less antibody production. So since here we found the deficiency in the antibody producing B cells and also the expression level of IL-21, it's not surprising to find that the follicular T cells in the Ph.D knockout mice was diminished after infection. This phenomenon was also found in both in the lung, but not in the spleen. Because in the spleen, if just like what we did previously, if we immunize the mice via the intraperitoneal immunization, it will not be affected. So it's airway-related follicular T cell impairment was found in the Ph.D knockout mice. And to find out whether this deficiency is T cell intrinsic or extrinsic, we just isolate the wild type CD4 T cells and the CD4 T cells from the Ph.D knockout mice and mix them before transfusion into a wild type mice. After immunization, we harvest the sample from these recipients, and we found that both the T cells from wild type or knockout mice can be differentiated into follicular T cells equally. So this deficiency is not T cell intrinsic. If not intrinsic, then who play a role in this event? Then we go back to the model. In the model, CD4 T cells need to be stimulated by the antigen presented by APC, and when these follicular T cells was generated, they need to enter the germinal center, and there they need to interact with B cells and the dendritic cells again. So you may realize that the dendritic cells play an important role in all this process. And in our previous story, we all know that the dendritic cells migration might be affected by the PLA2G2D deficiency. So everything seems to be linked together. When we isolated the dendritic cells from the knockout and the wild-type mice and compare their responses, we found that the PLA2G2D deficiency will resulted in the increased migration of dendritic cells from the lung to the drain lymph node, which is not new because we already know it. In addition to that, these dendritic cells also increase their expression of IL-1 beta. IL-1 beta is the pro-inflammatory cytokine, as we mentioned previously, and to find out whether IL-1 beta, whether excessive IL-1 beta will impair the generation or development of CD4 follicular T cells, we established an in vitro CD4 follicular T cells development model, and then we add the different concentration of IL-1 beta into the system, and we found that when we increase the IL-1 beta in the culture system from 1 to 10, it looks like it can benefit the development of CD4 follicular T cells. However, when we increased the IL-1 beta from 10 to 50 nanogram per ml, it will significantly decrease the CD4 T cells development into follicular T cells. So all of this phenomenon get an answer. The PLA2G2D deficiency increased the migration of IL-1 beta producing dendritic cells into the drain lymph node. In there, the increased IL-1 beta concentration will decrease or impair the development of CD4 follicular T cells. And this impaired follicular T cells development will cause the antibody producing B cells deficiency. So now to find out, to double check our hypothesis, we established a more complicated model. We transferred the dendritic cells from wild type or knockout mice, and we also treated the recipient with the anti-IL-1 beta antibody to modify the microenvironment of the drain lymph node. After this treatment, we found that yes, if we use the adoptive transfer of dendritic cells and also blocking the IL-1 beta, this treatment can reverse the antibody production and the follicular T cell development in the recipient mice. And also, this phenomenon was not restricted to the MERS-CoV infection. When we established the SARS CoV infection model in the H2 human H2 transduct mice, we also compared the antibody production, and we found that yes, in the SARS-CoV-2 infection, the PLA2G2D knockout also decreased the antibody production. And also in the human, because we cannot use the human to just challenge human with the different virus and then just knock out the PLA2G2D in expression in the human. So we use the in vitro model. We harvest some human PBMC and then obtain some macrophage and the dendritic cells from the myeloid cells in the peripheral blood of this human patient, and then we challenge these dendritic cells and macrophage with the poly-IC or the MERS-CoV virus, and then to compare whether the knockout of PLA2G2D with the CRISPR-Cas9 or the control plasmid, whether they could modify the status of these myeloid cells. And we found yes, the knockout of the PLA2G2D by CRISPR-Cas9 system can increase the IL-1 beta and even TNF-alpha secretion in the poly-IC or the MERS-CoV treated macrophage and the dendritic cells. So in summary, here we found that The Ph.D and Ph.D actually play a very complicated role in the immune system, especially in the airway immune system. The Ph.D deficiency will protect the mice from the acute coronavirus infection, but it may impair the generation of virus-specific antibody and follicular T cells in the middle-aged mice. This Ph.D deficiency caused antibody and follicular T cell deficiency could be reversed by conjunction of wild-type dendritic cells transfer and IL-1 beta blockade. So the origin factor is still in the myeloid cells. Although we now know about some roles of this signaling pathway, but still we found more questions need to be answered in the future, like to identify the Ph.D2 and DP1 induced signaling pathway, especially the role of pyrine domain in the regulation of the inflammation. Because we know that the Ph.D2 and the DP1 binding will decrease the inflammation production, but we don't know what is the mediated role or molecules, which molecule mediated this process. And we also need to illustrate the role of cells like the dendritic cell, macrophage, granulocyte, specific Ph.D2, DP1 signaling pathway in the coronavirus and influenza virus infection, and even some other respiratory bacteria or fungi infection. And we also need to identify the molecular mechanism underlying the Ph.D deficiency mediated modification on the follicular T cells differentiation. Another bunch of questions coming from the aging. We know that with the aging, the Ph.D and Ph.D2 expression will increase, but they are not the sole dancer in the aging, of course. So we want to identify the mechanism underlying aging-related lymphopenia and neutropenia in the SARS-CoV-2 infection. We also want to clarify the impact mediated by the aging-related lymphopenia on the host immune memory to the SARS-CoV-2 infection. And finally, since the aging is it's a complicated process, we want to know whether the change in the microbiome, oxidative stress, metabolism, and the senescence, whether this phenomenon discovered in the aging will also modulate the host immune responses to the virus and other pathogen infection. So finally, I would like to thank my previous lab, Stanley Perlman's lab, especially to my mentor, Dr. Stanley Perlman. He has been working on coronavirus for more than 30 years and made a very great and important and critical efforts in our understanding the pathogenesis and everything about the coronavirus. And also in this work, Dr. Kun Lee from the Paul McRae's lab and Dave Merholt's lab also contributed a lot to the whole story. And this work is supported in part by grants from the NIH. Okay, I will stop here and every questions about this story are welcome. And again, thank you for all well organization and thank you for your concentration. Thank you.
And thank you, Dr. Zheng, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you want to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So Dr. Zheng, we have some great questions already here and coming in. Our first question from our audience is, what is the mechanisms underlying Asapiprant mediated effects?
Okay, thank you. So here actually we have finished some like the primary studies. I also showed in several slides here. Looks like The first thing is that aspirin treatment will increase the dendritic cells migration from the infected lungs to the drained lymph node. We all know that when the virus infected people or the mice, the virus-derived antigen could be engulfed by the dendritic cells, and dendritic cells can play as a cargo to transfer these antigens to the drained lymph node, and where the dendritic cell will encounter the CD4 T cells, CD8 T cells, and then when the dendritic cells present this virus-derived antigen to the T cells, which could induce the virus-specific CD4 or CD8 T cells mediated immune responses. And we know that virus-specific responses is the best way to eliminate the virus without impairing the other host tissues. So yeah, the increased migration of dendritic cells is one of the mechanisms. But also we found that the treatment can decrease the accumulation of neutrophil in the blood and also in the lung infected by the SARS-CoV-2. And since the neutrophil has been regarded as a very important cells in causing the pathogenesis of the lung, so we are thinking that maybe in this model the neutrophil decrease is even more important mechanism. But still, we are now still working on that, and we try to clarify it in the soon future. Thank you.
Thank you for that, Dr. Zheng. Our next question is, our audience member asks whether PLA2G2D-PGD2-DP1 targeting therapy works on SARS-CoV-2 variant infection.
It's a great question. So we haven't tested a lot of different variants, but we believe that yes, if it works well in the SARS-CoV-2 mouse-adapted virus infection, it should also work on the other variants, because this treatment was targeting on host factors instead of the virus factors. So it should not be more affected much by the variants of the SARS-CoV-2. But yeah, of course, we do not have many evidences. We just tried 2 or 3 variants, and the outcome is very satisfactory. Yeah.
Thank you so much. We have time for one more question. Dr. Zheng, would PLA2G2D-PGD2-DP1 targeting therapy work on other respiratory infections?
Oh yeah, that's another good question. So yeah, after we confirmed the benefits of this treatment on the SARS-CoV and the SARS-CoV-2, we also try to find out whether it can work on some other diseases model. And the first thing jump into our brain is the influenza virus, because we know that besides the coronavirus, the influenza virus infection is another the great threat to human population. And every year we need to take the vaccine, and every year the efficacy of the vaccine might be different. And also compared to the coronavirus, the influenza virus can carry the more mutant, and so it's even harder to treat. And so we tried one influenza virus infection model in the mice. However, the outcome is not that good. So yeah, I have to say that in our model, this treatment, the Asapiprant treatment, did not work well in the influenza virus infection. It's kind of disappointing, but it could represent another important information is that so the coronavirus and the influenza virus infection are quite different. So maybe we need to find out some other way or other molecules to targeting for the influenza virus infection. But to some other respiratory infection, we don't know. We would like to give a try, and we maybe know more in the future. Yeah, thank you.
Dr. Zheng, I want to thank you for your presentation today. Do you have any final comments for our audience before we go?
Final comments, yes. So I think that there has been a lot of humorous efforts in the whole world that has been put into the SARS-CoV-2 infection investigation. And the first thing is that all scientists, all research staff would like to know how we can help the world, help the people. But the thing is that, so science is not like the magic. We need step by step to find the solution to the problem. And the vaccine is a big step. That it is. So we hope that besides the vaccine, we can find some other ways like the Asapiprant, like this signaling pathway, because we know that the mutation in the virus is always a problem. And we try to find another different angle to solve the problem, like we targeting at the host instead of the virus. It could be a good way, and the Asapiprant is not the only targets, the only potential targets. And we believe that there will be more and more other ways to jump in out. And we will be more glad to find out some other excellent outcome generated by the other labs. And we will also try to find some other way to fight against the virus infection. Thank you.
Well, I want to thank you again, Dr. Zheng, for your time. And before we go, I also want to thank LabRoots and our sponsor, Milteney Biotech, for underwriting today's educational webcast. And I want to thank our audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will be alerting you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.